In this series of short videos, we're going to take a look at what bacteria are, how they cause infection, and when you do get an infection, how antibiotics work to help treat that infection. But importantly, what happens when antibiotics don't work and what we can do to help prevent that from happening. So let's start at the beginning. Our best estimates are that the world is about 4.5 billion years old and that bacteria could be found on the Earth from about 3.5 billion years through fossilised evidence. But yet we've only known about the presence of bacteria on the Earth about 300 years through the pioneering work of people like Anton von Leeuwenhoek and Louis Pasteur, both of whom had the foresight to look for the invisible world of the microbes. But what are bacteria? Well, they're single-celled organisms that are much smaller than human cells. They tend to be simpler in terms of their characteristics, but they share a lot of the biological principles that human cells would have. If you imagine the smallest division on your ruler as being a millimetre, and the size of a bacterial cell is about 2 micrometers, which is 1,000 times smaller than that. If you were to look down a microscope at the cells, they tend to be quite simple in their structure. Some of them are coccoid cells, which are football-shaped, and others are more rod-shaped, which look like little baked beans. And often they can have appendages on the outside that allow them to swim and move around. If you watch the news, you might be forgiven for thinking all bacteria are bad when you hear about infections in hospitals and seeing foodborne outbreaks. But not all bacteria are bad. In fact, some can be quite good. But let's think about, first of all, what about Campylobacter? Do you think that is a good or a bad bacteria? Campylobacter is a bad bacterium. It's the #1 cause of diarrheal disease worldwide. It tends to be associated with poultry in particular, and that's why it's important to make sure we cook that food properly and wash our hands when we've been handling raw food. What about Staphylococcus? Staphylococcus can actually be good in some contexts and bad in others. It actually lives quite harmlessly on our skin, where it actually makes up the normal bacterial population on healthy skin. However, if the skin is broken by either a wound or a surgical procedure, these microorganisms can contaminate the wound and lead to infection. And in that context, it can be very difficult to treat. You might also have heard of an organism called MRSA, which is a Staphylococcus aureus that's resistant to many antibiotics, including methicillin, which means it's very, very difficult to treat. So now we've learned a little bit more about bacteria, about how small they are and about what they look like down the microscope, about how some are good and bad and indifferent. Let's have a look at how they cause infection, about how we can break that chain of infection, about how antibiotics work, and how, if they're misused, how we can develop antibiotic resistance. And finally, let's take a look at how pharmacists and engineers are working together to develop novel strategies for infection control.